Okay. Um, Good everyone. It's a nice opportunity to be here with you guys. By the name Kayode Rahim. So I'll be taking you on. So can everyone hear me clearly? So I'll not be. I'm speaking to myself.
Yes.
Yes, we can hear you.
Okay. Okay. Thank you. Thank you for confirming that. So really about I'll be taking you on protein structure and modeling in details. That we'll be talking more on homology modeling, talking about molecular docking and the like. So before I would dive in, so just to give you a kind of overview, that's what it's all about. Because if we are like, as we all know, we want to do a kind of research, so it depends on the domain you're working on. Protein is one of things we cannot do without it because either being gene, as we all know, this center of dogma. So we know from our DNA to RNA, from the RNA to protein. So the idea now is that okay, how can we get this about this idea of protein itself? What do I mean about protein? So which I will try to um, give a overview about it tonight. So let me start. So yeah, I just like to highlight like okay, what's protein itself? As we all know, it's kind of a biomolecule. It's large in structure. It's kind of essential. We can't do without it. And this is serves as its target. Either you call it enzymes or hormones or antibody itself. So as well, we have a some building block that bring about this protein itself, which are the amino acids. So all these points I'm making mention, they will be important in the course of this study because we need to know all this. So it's the building of this amino acid, and we know we have 20 amino acid. Though we have a particular one we work with, that's the 20 L alpha amino acid. So how are they being linked together? We have some specific bonds because these bonds are also important in terms of when we are trying to analyze our results, which I will show you in the course of this study also. So these protein are being bounded by this peptide bond. So you have a particular role they perform. And if a protein like this, because the main purpose here is to like tell you guys, okay, how can we do about the structure itself? So which I'm here to give you an idea about it. So the protein itself is more like it performs this function of the structure itself has a role it plays, and each structures have a particular function they perform in the body systems itself. So if a structure is lacking or is being absent, this bring about idea of this disease we all know we may have in mind in talking about. So this idea of how this protein come into play. So we have different level, we have different type of this, which I will show you as go on. So how protein are being made of? What are the okay, your screen is not moving. Oh, is it moving right now?
You can take it away from full screen. Yeah, it's moving
Okay,
now. It's moving now.
it's fine, right?
Yeah, it's moving now.
Okay, good. So as I was explaining initially, that this protein have been binding some particular bonds as peptide bond. Just keep that in mind. Then the combination of this amino acid that's 20, we have it bring about these types of protein, the level of protein structures. Then we have the aspect of the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. These names are just not there. They have their own role they perform because as we are going, because our main aim is to design a protein structure with the idea which I'm trying to explain. Please just follow along. So now with this amino acid itself is what I kind of is the starting point to build this protein. So in this amino acid that we try to what we unfold to bring about these secondary structures. So secondary structure, what is it trying to do? The aspect of the alpha fold, sorry, this the alpha helix and the beta helix shapes, like what you just on my screen now. They have like when you have the amino acids, they are in sequence. That's the primary structure of the protein itself. That's in sequence, in chain form. So if you see, can you see my cursor? Hello.
Yes, yes, yes.
Yes.
Okay, good. So now you see this line is not just a line. These are bonds in between different amino acids. So different proteins have different amino acids that bring about them. So the folding of this peptide chain that leads towards this protein folding that brought about these 3D structures. Because we cannot just work with this, though we can work with this, but it depends on our research interest. Let me give an instance now. Maybe I want to work with a particular target, a protein of interest. So I have the amino acid. I've searched all over. There's no available structure. What can I do? Will I say I will not work, do my research? No. There's a way around it, which I will tell you now. Because with the amino acid you have, we can able to what? To give a better reliable, a better 3D structures. It's not that because that 3D structures we need to perform our aspect of, let's say, okay, now drug design wants to design a particular inhibitor that will inhibit this particular protein that we call as our protein of interest. We cannot just put in the amino sequence to do so. No, we need to get these structures. At times, these structures are being made available on a database, that protein database, but not all are available. Some are like in the experiment space now. They get this sequence out, but the structure is not available. So what we need to do as a bioinformatician or as a biochemist or the like what is just like, okay, fine, you have the sequence. It can be maybe you're working in your wet lab, you have some protein of your amino acid sequence available, but you check on some database, discover that the structure is not available. So that brings a bad idea of, okay, we can design this structure ourselves. Though they are available experimental form, we can do so, which I will see later on, but they have their own drawbacks, which I'm going to highlight also. So what I'm trying to say in general, like these are level of these protein structures, starting from their primary structure, that is the sequences of the amino acid. Then the folding of amino acid leads these secondary structures. This structure is just having the alpha helix and the beta shapes. Then the multiple structure of this of the peptide bond now of this secondary structure that lead to this tertiary. That's what we need in performing our functions. Now, when you're not combining 2 or 3 of subunits of this protein together, then leads towards these coordinate structures. The example of this coordinate, like hemoglobin, is a protein, a oxygen-giving protein itself. So it's a combination of different protein structures that lead to this coordinate structure. Like now, as I said, some protein want to have multiple subunits. My cursor, this is one subunit, this other subunit. So they combine together to give these coordinate structures. That's how we got about this level of structures. So I've been saying this all around. So what are the bonds? What are the interactions? Because this interaction we need. These are not just forming on their own. There are some forces that are bringing these amino acids together, and they have their role playing. What they have their particular functions they are performing. Because all these things I'm trying to highlight, they will be necessary when we are interpreting our results. It will be so nice when you see some your structure. Okay, fine. This is what this one is trying to say. So let me highlight that for you right now. Now we have the covalent bond. What is it doing? This is a kind of the peptide bond I told you initially. Between those amino acids, these are peptide bond because the bond occurring there. What this is trying to do is trying to what? To form the protein primary structures. They form the protein primary structures. Then we have the word non-covalent. This is hydrophobic. That is, what does it perform? This is a weak interaction between these atoms. But what it's trying to do is to stabilize the 3D structures. It's to stabilize it. That's the role it's performing because we need it. Then also the hydrogen bond. These are like, because now let me just like revise you back to in our university days. Or now biochemistry, everyone is doing biochemistry or something like that. So we know we have the R group, we know R group. We have so all these amino acids are being grouped in these groups. So now the hydroxy group of a particular amino acid, when it binds to others, they form this hydrogen bond. For this hydrogen bond, they are needed. Why? Because they help in the stability of this protein. Because in our drug design, in our protein ligand interaction, these hydrogen bonds are highly needed, because these are what we needed to give what we call a scoring function, a docking score. When we get there, you understand what I'm saying more better. So these are idea we need to know, because all these bonds, they are needed to occur. If now let me give you an instance, because in the protein there's what we call active site and the allosteric site. So in the protein pocket, we are introducing our ligand, because the whole of that ligand, it can either be an inhibitor, either it can be to mediate or to stabilize. So whatever the role of that ligand we are introducing is having interaction with the amino acid of this protein at this pocket site. This is what I'm trying to explain. Now, if you see the kind of image I just tried to show you guys here, these are two amino acids, the serine residues. So if we look at the structure, because this what's it called now? The 2D structure of this amino acid, they have this the CH group OH. These are what we got an idea of these bonds. So if you can see this right form, we have the hydroxyl group and the hydrogen. By the time they bind, there will be release of water. In that sense, the hydrogen bond will occur. What it's trying to do is helping us to make sure that the ligand and the protein residues at that pocket site we're able to fix at that point. Now, what am I mean by this point? Now, if we are trying to introduce, okay, let me give you a case study now. We wanted to inhibit a role of a particular disease. We know that, okay, this protein are being the major cause of this particular disease. So now we are trying to introduce some ligand or inhibitors to inhibit it. What are we trying to do? We are trying to ask that ligand to be able to stabilize at that point, because if the ligand is still present at the pocket site of this protein, definitely it will perform its role well. But how can it be stable is the aspect of this hydrogen bond occurring there. If it's not fixed enough, definitely the ligand will just what? Will shift away. That will be there are some problems that now we're like trying to solve now, like what we call off target. Off target, what I mean like this, there are some, we introduce some ligand into the pocket site, but they get there, they don't bind. Why? Because most of the these bondings, this interaction are not occurring. So there's a way we also play with it, what we call optimization. We try to optimize the ligand again to fix that role. So these are all aspects of this, what I'm trying to, this interaction, this what I'm trying to guys to understand. So also, we also have the ionic bond, that's allostatic bond, that is interaction with the positive charge of this amino acid and the negative charge. These are the arrow groups. So I highlighted some example here for you to do. So what's it doing too also is to stabilize the protein structures. It's to stabilize the protein structure. Now we have the Van der Waals forces. What does it do? If you see here on this cursor, there's this green in this protein. This now look at this now. This is a kind of a sequence of amino acid. This is protein. This is a primary structure protein. By the time we try to fold it now, look at the folding. But how can it fold without not unfolding itself again? But there's a particular role of this weak bond. That's Van der Waals bond is playing that role in that aspect. It was to be able to compact it and make it stable. Am I making some points? Hello?
Please, yeah.
Okay. So yes, just like no, if you guys are okay. So now I'm trying to guys to know. So now, how can we now determine this protein structure experimentally? We have this known method, and that's the X-ray, the nuclear magnetic resonance spectrometry, and the cryo-light electron microscopy. But they have limitations. Though they are good enough, and the limitation is that these are being existing in days. Like, you want to perform this experiment, you need a lot of money and it consumes a lot of time. That you want to, like, okay, fine, you have subsequence, you want to get a 3D structure protein. This can take you days, more than, and a lot of funds will be there. But the aspect of this computational stuff now, it has made it easy for us to, like, within days, 1 day, 2 hours, depending on your computer resources you have, you get all these things done, and we are good to go. So what are these X-ray doing? It's just like you have the crystal structure of the protein itself, you introduce it, the X-ray will the kind of the what am I saying now? Like, it's producing a diffraction. This pattern will be the one I give. Okay, this pattern is giving me. This will likely be the electron density. It's a calculation. But in this case now, the limitation is that we will need a high quality crystals to perform this X-ray. That's one of the limitation it is. Then also the nuclear magnetic resonance spectrometry is also, it's a good one, but in this case now, how does it how this do is kind of predicting the structure is kind of a distance between those amino acid residues atoms. These are calculation. These are experiment they do so. So I'm just trying to summarize this for you. So now also we have this the camera electron microscope also, but this is kind of more computational intensive. Like, what I might think, like, it's more expensive to run, and it have a lot of, you can see it has required expensive equipment and advanced computing stuff to do this particular cryo-electron microscopy. But how can we now overcome this limitation? So simple. Now that's the idea I want you guys to introduce this computational stuff to do this easily. That idea of this homology modeling. Okay, so what am I mean by homology modeling? The word homology means something comparative, to compare and contrast. So let's start now. Our major aim is to design a 3D structure from our amino acid sequence. So with the help of this model, which is being done, and these are done on computer easily. So we have some available resources, web server, software to do so. So now, this homology modeling simply means, I say, it's comparative. We are trying to compare. How? What about we are comparing? Now, we have some already experimented 3D structures already available. So we have our own sequence of interest. That's the query sequence now that we want to design 3D structure to do this. But what are we trying to do now? We are going to compare with what is available as experimental one. So those that give us a better template will be our next action to work on, which I'm going to show you guys. So, but we have to meet some criteria for us to choose a better quality models. So now, if you see, I write here that it said, if similarity within the target sequence, that is our query sequence now that we have, that we want to design 3D structures, then we need a template like, okay, fine. You see the kind of image I show you as explained, it's quite all right. We have the query sequence. This is our sequence, and the homology sequence. This is one available, the experimental one. So by the time we run it, we will generate series of templates having this similar sequence. So we now have to choose the one that have the best sequence similarity. So what are the points we need to look out for? That's why I said, in general, 30% sequence identity. That is one that have within, now we have our own sequence we are trying to work with, and the experimental sequence available. By the time we compare the software or the server I'm going to use, it will generate a lot of templates having this same sequence similarity. Then we need to choose. So our cutoff point must be that anyone below 30% will not be a good model. But that doesn't mean we cannot use it. That we can also use it, but using other way now. That's the other way we're going to do that. Let's say now, let me give a case study. Okay, after I have my sequence, I try to introduce to whatever server I want to use, and I saw that the sequence was giving me sequence similarity or sequence identity below 30%. Doesn't mean I will not continue. No, I will definitely continue, but it doesn't mean I have to seek for other alternative. And that alternative is like using what we call profile template, or what you use what we call hidden Markov model. These are like a machine learning models to solve this problem. So what would that done would do is that it's going to like it's query the server again, give us similar sequence that have this similar sequence that we're to work on. So that way we can also work in that aspect. So I don't think you guys getting it. Okay. So that's being said. So we also have another way that we can also design 3D structure, but right from the word from the amino acid sequence only. We don't need templates. We don't need the experimental one. Are you getting my point? That we're just working de novo sequence. I'm working from the main source now. Like if I have my sequence, I'm not maybe the experiments of or that particular have not been done before. Doesn't mean I will not know. This way we can also do so using what we call ab initio protein structure prediction. But it has some drawback. What are the drawbacks? At least you have some number of amino acid sequence that you can work with this available web server or software we are using. Though there's some software we can use, but they are commercialized. What do I mean by commercialized? That is, you need to pay to have access to it. But they are also available where easily available for someone to use. Now, if you see on my screen, I'm showing you two major web page. We can use this ab initio projection, but the limitation here is that they can only take amino acids that, like this one now, this variegated this, it can only take amino acid within the range of 100. So if it's beyond 100, it will accept it. So what this one is trying to do, this is a kind of automated workflow. We don't need to do much. Just copy your sequence, whatever you have a sequence, but make sure that your sequence you have in mind or interest or target or query must be less than 100 sequence. This the amino acid sequence and what I mean now, so that you're able to do so. But a lot of people are also using same server as the time you are also using. So the result may not come out immediately. It may take an hour, some 2 hours, some 3 days. It depends. So that this works. Then also we have this one, but this one can take up to 200 amino acids at a go. Then you just put in your details and sit back and relax. It will generate likely structures having that same similarity. The main principle here is that this server they work on some experimental sequence available. So it's like you're trying to query the server, whatever it is, trying to generate those sequences that have similarity with what we have. So to bring it out. But the limitation I told you is that they can only take 100 amino acids and some 200. While we have some commercialized software that can take more than that. I highlighted them here also. We have the Gaussian, we have the games, we have the Amber, and the CP2 case. So these are like softwares, but they are commercialized. They are available to work with. Also, we also have this Galaxy Web to do so. Let me just show you so that it will be easy. Can you see my screen? Hello?
Yes, we can see your screen.
Okay, thank you. Okay, so just want to like show you what I'm trying to talk about. Now this Galaxy Web. So it's so easy. Now what you need to do, okay, let me just show you, let me just particularize one. It's time permission so we can able to cover other aspects easily.
Please try reshare the screen you want to. You can only see the slide.
Oh.
Yes,
You can also
try to reshare
the.
Yeah.
Okay. Let me. Okay. Can you see a web page?
Yes, we can see.
Okay.
So when you want to switch back to the PPT, you can just reshare the screen again so that so
Okay,
you can.
okay, 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 okay. Let me just go there. Okay, you can see the screen, right?
Yes, we can see the Galaxy web.
Okay. So just to like show you what I'm trying to explain now, if you see closely, you see the sequence. Now it's showing you 30 to 1000 amino acids. So this means, which means this one can take up to 1000 amino acid sequence to go to run. But we need the word, we need the word, the faster file to run this. Okay, let's quickly just show you what all these faster file are. Okay, can you see my screen? I'm trying to get a sequence. Do you see a sequence?
Yes.
Okay, so this is the sequence that I worked on. So now let's try to where are you? Okay, good. So you copy and paste. So now we need the job name, whatever the job, the name of your sequence you are working on. So this particular sequence is on P53. So I just put it there. Optional, you can put your email. Why this email is needed? Maybe so that when the result is done, you can able to easily access it. Because now look at it now. On the left side, it's showing the email average runtime is what? 7 hours. I told you. Because you are not the only one using the same server. So people are like, others are also working on it. So that's why it's good to put your email. So by the time the job is done, you'll be notified. You just click on the link and it will bring it out. So now if I put my email, okay, sure. Okay, so just hit the submit button. Okay, can you see now? So it's been running. Now by the time it's done, you have your template out. Do you get my point? Yeah.
Yes.
Okay, good. So let's go back to where's our slides? All right. Okay. Good. Okay. So that's about ab initio protein structure prediction. What I just done is that I just need the amino acid sequence only. I don't need anything like templates. But if we are using the aspect of homology modeling, because what homology means to compare, we need the templates. Because that template we need to what? We need to blast. Like I don't know if you have guys, I think you have thought about blasting. So we need to like do kind of sequence alignment, either use multiple sequence alignment or the pairwise sequence alignment to do so. That's the aspect of homology modeling. But we have some web server that all these are being embedded. We don't need to stress ourselves. Just put in there, which I will show you also, is to perform this. What I'm just trying to say, the sequence alignment, to align it is to generate the template and give what we need. So that's that. So now let's go in the details about homology in particular. Okay, so these are the steps. These are the steps involved in homology modeling. So now, this first step, it's required. You see, I highlighted 1 to 4. That is the choice of template and initial alignment. Now, what am I by choice of template? I told you we have our sequence, the amino acid sequence now. It can be more than 1000. It depends on what you have. So what we need to do in this aspect, we need to the amino acid, then we need to do alignment. The alignment can do either use, there are some MCBI database that have this alignment, either use cluster W, omega, irrespective of alignment you want to do. But we don't need to stress ourselves. We have some web server that make everything so easy for us to do. You just sit down and input it. It's a kind of web server user friendly when you know how to put in there. Then we have some that we need some command line to do so. If you are a bit good in using some Python language, they can also do so in that aspect. That's AlphaFold. So in that one, because this will generate more and more high quality 3D structures. because it will be like generating structure that will have less loopholes. You see these loopholes or something, you have some side chain, because these are necessary for a protein structures, because they be needed when you are doing your molecular docking, your MD stimulation, whatever you want to do with your protein structures. All this will be needed. So in this choice of template, this when you blast it now, because you are blasting against, it will generate some sequence that has similarity to your sequence of interest. So these are like, it will generate some template. Is that template we need now to continue the next process. Now, by the time you get your template, there will be some errors also. What are these likely errors? That is what we call this alignment correction. Now we are trying to correct where there be mistake in the sequence. Maybe it's not giving us what we expect to, so we can correct this one manually. We can correct all this one manually. We can be done that manually also. So now when that is done, the next thing for us was to generate a backbone generation. To generate the backbone of that, after this alignment is done successfully, so we need to generate the backbone of the protein itself. What about the backbone we're talking about? That's the aspect where the secondary structures, I said earlier on, I don't know if you guys are paying attention along. This backbone is not needed. That way the aspect of secondary structure, because now initially we have our primary structure, the sequence. That what we have initially, and we have generated template that have similarity to what we have. So we need to now fold it to be coming to form of a secondary structures. So that the backbone we now have the need of the alpha helix, the beta sheet. So these are needed. So this aspect is not done on this backbone generation. So after that is done successfully, loophole modeling is needed, because they play a crucial role in the protein function. What do I mean by this? The loophole is that he's talking about gaps. I don't know if you guys have this bit experience when when you have a sequence alignment, you see some gaps in between your sequences. These are loopholes that we need to model, we need to correct. So this loophole modeling is needed in this stage now, after you've done the backbone, because if these loopholes are needed as are present in your protein, it will be a problem when you're working to other analysis. Though we also have a way we are correcting this with some software. If you are performing molecular docking or MD stimulation, we can also correct this. We can also correct that easily. But to have a good job done from the right onset, you can do this and correct it yourself. Because if this loophole, if this gap are present, it will affect the function of the protein. It will not give us the real function, what this protein is doing particularly. Okay, so that's that. Then the next thing, what is side chain? We have, in your protein, we have the C terminal, we have N terminal. Because this side chain are also needed, because all these things are needed for our ligand to bind. Some ligand may not bind rightly at the pocket side, some will bind at the far distance. And when we have this allostetic side, these are far from the active side of the protein. Because now in drug discovery area now, research are now working towards targeting allostetic side. Why? Because they are trying to limit the aspect of these off targets. Because you introduce some ligand into your pocket, into your protein target, into your protein active side, they may not bind. They will not be there dangling around because they are not performing. And when they don't bind, definitely that ligand of interest is of no use. So what can we do to solve this problem? Okay, what can we now target off target? Like let's target some other side apart from active side, that will idea of allostetic side, which I'm working on that research, and which is giving us something good enough. We cover that those at the allostetic side, this ligand are able to bind firmly and able to perform their role. These are done easily. So that being said, now we need to optimize our model. Let's say because after our model is being built, we need to optimize it. Because why we need to optimize it, we are trying to get a high quality and high profile protein 3D structures. Now, there's what we call Ramachandran plus scientist. Ramachandran is a scientist. He did an experiment working on protein. So he's a protein chemist. So this Ramadan flow is what we needed now. Is where we'll be checking out some parameters. Okay, how many amino acids are in the favorable region? How many amino acids are not in the favorable region? How many amino acids are in the favorable region? So because this thing we need to check for. So if we check now that okay, I've done my modeling successfully, side change successfully. Okay, let me check if my number of amino acid. Okay, let me quickly show you so that will be more better. Let me share my screen again. Okay, so can you see a paper?
Yes, you can see.
Hello.
Yeah, we can see. We can see.
Oh, okay, good. So this one I'm trying to explain what I mean this transverse plot, because that's what the modeling optimization come into play now. Now, what is this graph? What is it trying to show? Okay, let me zoom it well. Is this clear enough? Okay, so now you can see it in the first quadrant. So it's showing us the residue. These are the amino acid. These black dots, they are the amino acid. This is a particular protein. This one protein now. Because after we have done the modeling, we are able to correct all possible where we thought. We have tried to correct the loopholes, but still we need to confirm our model, the structure. Is it fit enough? Now, if we check closely, we have some amino acid. These are the amino. This black square, this small block, that the amino acid. Now if we check the ratio, that is, we have the residue in the most probable region. How many percentage of it? 86, which is a good model. Then the addition that is in the allowed region, how many are they? Also in 11.4%, which is a good one. Now we look down, I have residue in disallowed region, which is what? That means in this our particular protein, none of our residues are found in disallowed region, which is a good one. So we can move ahead. These are done with some web servers to do so, which we are going to particularize and check easily. Okay, let me go back to my slide. Okay. Okay, so I guess I'm on my slide, right? Hello.
Yeah, on this slide, yeah.
Okay, good. Thanks for confirming that. So that's why we did this model optimization. Now we need to validate. We need to validate our model. We need to compare what is available. Then how can we validate? Now we need to go into protein data bank to check some available structures that have that similarity of our model. So we may find, definitely we will find. If we won't find, that doesn't mean that we cannot proceed, because with this all the criteria being said, it's done successfully. So now it now depends on what you want to use your protein structure to do. Either you want to use for molecular docking, either you want to use for molecular dynamics, and whatever other maybe you want to use for protein interaction or protein DNA interaction. It depends on what you want to use for, because you are able to get a 3D structures of your protein successfully. Okay, so now this molecular docking is done using the protein. Now maybe we have some libraries of compounds we want to perform. We can easily maybe molecular docking to check okay how stable the is when we are trying to run the simulation and the likes. Okay, we are going to work on this molecular docking easily in our next class. So now that being said, I'm talking about database. What are some database? What are the protein database we have? We have a lot of them. So now we have the protein data bank. I was trying to explain. You see, I tried to get an image of it to show you what it is. These are like a repository having experimental 3D structure available. So we have more than 190,000, and it's being deposited daily and daily. Even you as a scientist or a researcher, you can also come up with your own protein and deposit there. People do it. If you have what you see you have is good enough, deposit. That's what people are doing. Maybe you are working on a particular sequence, You can do the structure and deposit. People are doing that also. Then we also have the class. That's the class architecture topology. So it's also, these are resources. Now we want to understand now. You have the sequence. You don't know what the function is. You only have the structure, but what's it performing? So we have some website where we can also do that to determine the likely function of that protein, and where the family, the evolution, where is it coming from. So we have some website that can also do that for us. So we also have groups as the structural classification. We're trying to let's classify down. Let's classify in terms of families. Let's classify, okay, it belongs to so family. So these are done also easily for us, trying to check the evolutionary, where it comes from. Then we have this good data that's Uniprot. It's so common. People use it very well because most of the time, I use it most of the time because if I need a sequence, if I want to know a particular, so it has a lot of information on this that we can do. Okay, let's try to explore one. So then we have the Swiss model. Okay, if I move on to that. So like I said this is a kind of okay. So Swiss model, this is a kind of automated one. We use this for this homology modeling. Now, all the steps I highlighted initially, we don't need to do it one after the other. This Swiss model is a kind of embedded in. It's already automated. You just input your sequence, then sit back, and you'll be seeing it. It's be loading, like you'll be performing the sequence alignment one after the other easily. So we're going to do a practical session on that to see what I'm trying to say. So what we're trying to do now, we are trying to start for a kind of 3D structure of that particular protein itself. It will suggest some series of templates for us to do that in that aspect. Then we have Raptor X, we have AlphaFold, we have a lot of protein 3D structure projection that we have, but they have their side drawback aspect. One is good, one other, but the best one are currently, it depends how good you are. AlphaFold is good. Why I say so? Let me share an experience I have. One time we went to a conference because that's what the researcher asked me. Okay, because I did the project on this particular modeling. So, but I use Swiss model. So one of the professionals asked me, why can't I use AlphaFold? This is a question they will ask because these are like, it's good for you to explore all the software that's available, start to what, to confirm your results. You don't just need to sit on one, okay, I've done this, okay, this are good. No, try to check other softwares. By the time you check, because after I discover I use that AlphaFold, then I have a lot of template aside from what Swiss model gives to me. Not that it's not good enough. I might make some points. So that's why it is good to explore other resources to ascertain your results. So good. Okay, let's quickly just show you interface of all this one. Let me just copy this. Okay. Okay. Okay, can you see my screen?
Yes.
Okay, good. So now this Swiss model, you can see the interface is so friendly, so user friendly. So now you just need to start your model, but we're going to do the practical session easily. Everyone is going to practicalize it easily. So just to show you how it is. This is the, once you click on it. So let me just explain this. I may not. So if we look closely, we have search for template, build model. Now, these two things have different meaning. Search for template, that is, you want this website to generate a lot of templates for you to work on. Okay, I think I did one. I will show you. Then this build model, that is, you want it to do the whole prediction for you. You are not selecting. You get my point now. If I click on this build, if I have my sequence, I paste my sequence here, then I click on build model. That is, it will not build it for me. I won't able to have access of, okay, maybe I want to select a template best fit or something. I won't be able to do that if I click on this build model. But if I click on this search template, then I have the option of, okay, selecting. Okay, I think I did one. Okay, let's see if it's somehow. Okay, good. Now, this is what I'm trying to say. This is my sequence. Now you can see, let's look closely. You have the summary, you have the template, we have the model. Now, this is my summary, this is my template that I was telling you about. I have about 50 templates. So this template, they have their sequence identity. Now, let's come down and look at the template results. If you follow me, you see what I'm saying, that now these are the templates with different PDB ID. That if I click on this ID, it should direct me to the protein data bank. What are the PDB ID? What you are trying to say? It's trying to tell me that, okay, these are likely sequence that this is my sequence now. This one I highlighted now. This is my sequence of query. This is my interest. Okay. Now I've introduced it. Now this website give me some likely sequence available already. They have this similarity, but it's now categorizing them based on what available similarity. If you check at the first one, that is having 100%. That is more like a kind of no. It's like exactly the sequence I'm working on. This one, I get my point. Likewise, this also. Then look at this one now. You can see 95.6. That is not. It's more like it's not like it's giving us percentage similarity of the sequence itself. So if you check now, if I click it down, you see. Let me just, okay, good. You can see these are the least. It's up to 50. Okay, let's just check down. You can see these are up to 50, and they have different sequence identity. Now look at this now. Look at this. This is telling us it is the sequence identity with what I want to work is 0.48, which I can work with this. I can. So now if I want to select, I will select up to like, okay, no. Okay, sorry. This is what I'm trying to show you. This identity I'm talking about. You leave this. But I will get to the main part. I will explain what I mean about this. These are like factors we need to check. So now this is the identity. I'm trying to explain how similar, because the homology is like telling us we are trying to compare now. I have the experimental sequence already, then I have my own query. So what are the similarity? The similarity between the experimental one and my query one is 100%. That is, they are like alike, no different. Then I will go for it. But what I do in my own case, I will select 3. I will select 3 of it. So it's telling you the method. What method did they use to determine the structure of this one? As I explained earlier on, I told you we have some experimental one that I highlighted those 3 methods for you. If we look closely now, this is the MRL, this is the Caroline method. We have the AlphaFold, we have the X-ray. We can see X-ray. These are the methods they use to determine it. Okay, because when you're trying to report your research, you need to say this out. Okay, now if I click on this, because I'm selecting this now because it's already clicked now. This is structure. Can you see? This is structure already. Okay. If I say, okay, no, let me check the other one. The other one, it's different now with different PDB. This is the PDB ID. Because by the time you click, go to, okay, let's do one more. Let me just, it's good to, okay, I'm trying to go to Police Data Bank. Okay, good. So let's try to copy. Oh, what's that? Okay. So now this is the PDB ID. Let's copy this and see. It should tell us. It just give us the main information about that structure itself. Okay. Oh my God. Sorry. Okay, good. So then I paste the ID. Allow it to load. Okay, good. Can you see it? Is any different? No. Now it's still not the words. You know, I told you I was working on PDP 53. Now this is the white type. This is the structure of it itself. So are you getting what I'm saying now? This is the method they use. This is what electron microscopy is to do so. Now the resolution. So and like that. So that's how it is now. It has no mutation. Which organism, Homo sapiens. So these are the information we need. So in my own case now, I may not choose this protein. Why I will not choose? Because of the resolution is so high. For a good protein to work with, we choose at the range of 2 to 3 Armstrong. Are you getting my point, please? Hello? Hello?
We are with you. We are with you.
Okay. So I was explaining that because these are a parameter why we need to select our protein. Now we have generated a lot of templates to work with, a lot of it, but it doesn't mean we need to work with all of them. No. Like this, in this case now, we will not go with this. Why? Because of the answer is so high. Okay? The normal, like, it doesn't mean you cannot work with it, but you need to have something good enough. At least we prefer something range of 2 to 3. It's okay to work with. Okay, let's check the other template. Now look at this also. This is another one. Okay, good. You can see the structure of it. It has a similarity of 100%. This is also another one with different methods. So by the time you now click that, these are templates we have now. By the time we click on this to build the model, it should build it. That is, it's going to model it at, like, to this particular experimental protein that we have. So that's what information I'm trying to pass across. So let me just okay, good. So okay, so we stop there for today. So we continue. If you have questions, so that I don't want to too much bombard to our permission, so that we can able to. Okay, good. So, any question? Okay. Hello.
Hello, Mr. Carol.
Yes, I can hear you.
I have it. Yeah, thank you.
Okay.
When you were presenting, you talked about Swiss model and that you presented the work in a conference, and one of the listeners asked you, why didn't you use Alpha Food? So what
Yes, yes.
what was the rationale behind this question? Is it that Alpha Food is better than model or what? And
No.
in what way is Alpha Food better than Swiss model?
Okay, good. Thank you. No, that is better. Now, why she asked that question is that
Okay.
AlphaFold is like a deep learning model. I get my point. What AlphaFold is trying to do is trying to generate more templates. Okay? It's trying to generate more templates to work with. So that's why he asked me to why can't I use AlphaFold? Okay, it doesn't mean Swiss model is good enough. It's good enough. But AlphaFold is also like a more recognized like how I call it now. You know, like not a web server, but a command line to work with to in terms of this 3D structure itself, because it generates a lot of templates. You have an option to work with, so that's why.
Okay, okay,
Yeah.
okay. Then also, to add to my question, the last time I navigated Alpha Food, I look at it. It's more or less like they've already modeled those protein there. You just for you to pick, right, to search and pick, right, or you can
Yes.
initiate a modeling of a protein.
Yes. Yes.
Okay. No.
Yeah.
Thank
Yeah.
you.
You're welcome. Okay. So more questions?
Hello, sir. Hello,
All
sir.
right. Go on. Go on. I can hear you.
The question, I don't understand the question he asked last. So the answer you gave him, I don't understand. The answer you gave his last question, I don't understand. I'm interested in his question.
Okay. You asking about upper food, right?
No. For initiating.
Okay. Yes, sir. That's my last question. Was that can we initiate just like in Swiss model, we input our template sequence and initiate a modeling algorithm. Can we also
Mhm.
use AlphaFold for that too?
No. In this alpha fold is not like that. It's just like you just input in your what's it called sequence into your the web server. Not it doesn't work that way. Okay.
Okay. Yeah.
Yeah, it doesn't work that way.
Thank you.
Yeah. Okay. More questions? Thank you for the submission. Is sweet not limited to a particular protein or does it work? No, it's work with any protein. So far, it work with what we do. It will query like it will like you are doing now. The sweet model, as I told you, is an automated one. That is, all the steps are highlighted initially is already embedded. It will perform this alignment. It will do even though the sequence there's no like a similarity, but it will still continue to search for you using this ID COF model or profile analysis. So these are embedded already. By the time we do the Pascal, you see, I will show you when it is running. You see what all these points I'm trying to explain. Just to like prepare your mind so that when we are doing the Pascal session, you're able to follow along that okay, this is what I'm trying to explain.
Okay, all right, sir. I
Yeah.
have another question. Maybe it is not question. Since Swiss model is automated, so why do we have to go for other web server since we can just paste our sequence and it generates it automatically? So why do we have to go for another one since it is automated?
Okay, now, thank you for that question. Good question. Now, why you need to go for other one is this. In research, we don't just like okay, since you are aware of now, don't forget we have don't know we have Swiss model. We also have Raptor X. We have Galaxy Web. So all this one I may mention, they are good enough to work with. Okay, they are good enough to work with. So to make your work so innovative, someone good enough, you can implement. You can just apply three of them. I don't see where I get my point now. Let's say now I have my sequence now. Okay, I will not just base my work only on Swift model. I can just okay fine. Swift model give me my result. I put it down on my paper. Then I can also work with what AlphaFold to like supplement what I have. Okay.
Okay, all right, sir.
Hello.
Oh, yeah, we get you.
All right. Any more questions?
Okay. Hello, sir.
Yes.
Can you hear me? On other
Be-
software, on other pipeline for modeling, the steps you listed, is it for everyone aside Swiss model?
It's the same.
You have
They
to
follow
follow
the
the
same
steps.
step. Or the step is general. They follow the same step.
No, actually, I have not yet understood the step. I think you would, in subsequent class, you will explain better.
You don't get the step yet.
Yeah, yeah.
Okay, let me
Yes.
quickly go back there. Uh, let me get the screen again. Okay. Okay. You mean this, right?
Yes, sir, exactly.
Okay. Now, as I said earlier on, homology is like we are comparing now. So most of this what I may mention, they follow these same steps. Okay, but the way they optimize, the way they work with is like now different. Okay, now I said this choice of template, it will generate a lot of templates like having similarity with our sequence. So all this, how all this template has been done is kind of do what we call alignment. It will align it, then check. You know, I don't know you guys, we have done something sequence alignment. You know, this multiple sequence or pairwise, right?
We've done it. Yeah,
Huh?
yeah, we've done it.
Okay.
We understand.
Okay, good. So when this sequence, when this alignment is done, so it's trying to check, you know, is it blasting it now again one after the available sequence available. So anyone that have similarity, it will like save it, you know, it will come to save it out to its own um database. So once all this has been generated, so that's why you have that 50. That is okay. He has queried the whole available experimental sequence. Okay, so now you could really get 50 that have similarity. All right, so that's the template on that first step. So now we now have to choose. Now we choose checking for some parameters. Okay, what are we checking for? I you know I remember one point checking for the resolution. Now we check. Okay, he has given all these templates in this particular sequence. He has similarity with this one. Okay, we need to check. We don't just stop speak. So how are we going to check now? Is what we call this sequence correction. If there's any kind of error, we correct. If it's not, we let it go. We choose the better one. So that's aspect of this alignment correction is trying to do. Then this backbone I told you is a kind of building now. We have choose a model now. This backbone is like secondary structure now. Is a sequence already. You know, we want to build a secondary structure checking for the ellipse and the beta stuff itself. So that's it done. So the loop modeling also we check for gaps in the sequence. Is there any gaps? Is there have gaps? We need to correct that also. We need to correct that. So now what I'm saying that there's some software that you all this now I'm saying you'll be doing it yourself. But in case of Swift model, you don't do all these things now. I get my point.
Yes, sir.
Hello.
We are with you. We are with you, sir.
Okay, good. So now that's what I'm trying to let you know that you see all these steps are like most of these software are built on these steps, but some we want to do it yourself. Like you check one after the other. If there's any one that is not conforming to what you want to do, you correct. But in the case of Swift model, that's why it is being written automated. This is done exactly. Now, you will see from here now, you see homology and you see the model building. So what Swift model will do for you, it will take from this from choice of template you choose. After choosing, then the alignment correction, then down up to this point is done inside, like embedded already. So what you need to do now is the model optimization checking for some parameters. Okay, is it how many amino acids are in those ready in those region? How many are they? So these are the model optimization you need to check yourself. But the rest, this one is done within the server itself. Do I try to
Yeah,
you get
yeah,
my
I
point?
get you. Yeah.
So by the time we do the particle section, you will see much what I'm trying to say.
Okay, thank you, sir.
Yeah, you're welcome. Okay. Any more question?
Yes, sir. Another question, sir.
Okay.
Since we build our protein ourselves, we
To
build it
be
ourselves.
what?
Can't we use it directly instead of finding templates that make us go through this kind of alignment? Can't we use
Sorry.
it? Because
Sorry. You
hope
just
you
repeat
get my
yourself. Sorry.
question.
You repeat yourself.
Hello.
Go on. Go on. I can hear you now.
Okay. What I said was that since we built our protein ourselves for a particular thing, why can't we use it direct instead of blasting it, checking for templates? Can't we use it ourselves since we built it for a particular aim? Instead
No.
of
No.
us going
Okay.
through this, checking for another person's um, sequences, can't we use our own
No. No.
sequence direct?
No. Okay. Okay. Now this voice. This I get a question. Now you are saying why after you build the model, why is it checking for other one, right? No, that's not
Yes,
how it
yes,
works.
sir.
That's not how it works. Now
Okay.
this like we are checking is a template. It doesn't mean your model you are building is copying or something. It's just like a template you are working on. So we are building a model based on that template. Okay.
Okay.
So it doesn't mean you are like copying or getting someone's work. No, it has brought a lot of templates for you to work on. Okay, you choose. Okay, let my model I want to build. Let it build on this template because you need a template to work on. If you do want to do so, then you cannot go to aspect of ab initio. You start from scratch. You don't want to use any template. I get the difference now.
Okay.
Homology modeling you need template because you deal with comparative. You are trying to compare. But ab initio you just get your amino acid sequence. Then you introduce it. It will give you structures based on that sequence. Is it clear?
Okay. Yeah, yeah, it's clear, sir. Thank you very much, sir.
You're welcome. Okay. You're welcome.
All right, thank you so much, everyone. So, and thank you, sir, for the wonderful presentation. So we calling it a day, and we'll be meeting, God willing, on Thursday for the practical session of all the parts that have been discussed.
Okay.
So,
So
and okay,
sorry,
okay.
sir.
Yeah.
So if you have some sequence, you can just come against next class, so we can just work with. So I will not be working what I have. That will be more okay now. Okay, share line more on the parameters we are need to look for while we're collecting. Yes, I will. I will. You will surely see that. I will explain that in details also. So just to like prepare you because when we are doing the particle section, so to get the main aspect, the parameter is not that is what I've explained initially. So what that parameter I'm talking about is how many residues are found in the allowed region, how many residues are not found in the disallowed region. So we need to take this in place. Then we check for the Z score. So you see all this also. So it's not something much also. So what I want to say that if you guys have a sequence in mind, bring it along, then we do it along. So we work together. That will be more better off. All right. Thank you.
All right. Good night,
Thank
and have a nice night's rest.
Yeah.
thank you.